Okay, hello everyone, and welcome to Module 5 of our Single Cell Bioinformatics Bootcamp. Today we're going to be talking about advanced topics in single cell analysis. These are the things that you might do after you've already got your UMAP and your clusters and your cell types assigned. These are the sorts of things that can really bring out the best in a data set and really allow you to dig deeply into the biological meaning behind your results. So let's talk about what we've done so far. So essentially, so far, I've shown you how to do the basics of loading the data, doing basic QC, and then normalizing and scaling. We've then talked about how to do nearest neighbor graphs, principal component analysis, clustering, and low-dimensional embedding, such as t-SNE and UMAP. We've also talked about a couple advanced topics now, such as cluster marker analysis, cell type identification, and cluster DGE. But we're now going to step into a few more, even more advanced topics, including cell cycle analysis and trajectories. And one more thing, which is not even on this list, which is just generally how to build module scores. So that's the main thing we're going to do today. And then we're going to touch a bit on how to integrate data sets, which is just something that is not even necessarily an advanced topic, but just an important practical skill to have when you're doing single cell. So like I mentioned, one of the things that I really want to do in our final session next week is to go over anything that I've missed that people really wanted to see. So if I've missed anything and you really wanted to see it, just go ahead, put it in the chat or shoot me an email at any point before next week, and I'll include it, as long as it's not going to take the whole time. All right, so with that, let's now jump over to our Module 5, R code. So hopefully these installs have gone well for you. If they haven't, feel free to put any errors or any warnings in the chat. This can be really, when you install from GitHub like this, that can be really hit or miss. And so if you have a question, there's a great chance that everyone else has that question too, or at least a lot of people have the question. So hopefully this worked. And if it didn't, no stress. So you can go ahead and highlight all of these. If you didn't actually do the Monocle 3 one, then you can just skip it and just do the other ones. But I actually do have Monocle 3, so I'm going to load that up. All right, so we built this function last time, so I'm also just going to include this as kind of a preliminary. This is a method for getting a color palette that we're going to need later. So I use this here. If anyone has any questions about how this is working, I'm happy to explain. But I think for right now, I'm just going to gloss over it. We get our databases for enricher, and we set a random seed. The reason that we do this is because some of the things we're going to be doing today have a random number generator component. And just to make sure that you see the same thing that I see, I set a seed here. And that way, your result should look like my result if we all set the same seed. That's not always true, but fortunately, that's not going to be that's going to be true for at least the things we're doing today. All right, so let's go ahead and recap what we did last time. So we got the PBMC data. Go ahead, grab that, untar it. We're going to go through our pipeline that we've now talked about a few times. And the only new thing we're adding on top is this renaming the identities. So these are the cell types we figured out last time, the cell types that correspond to each cluster. So we go ahead and run this, and it just goes all the way through the process. Really doesn't take terribly long at this point. Okay. And just to reiterate, we run these. Does anyone remember why we include this line on 87? Does anyone remember why we did that? Feel free to put it in the chat if you remember, or just unmute. Okay, so the reason, oh, okay, yeah. Sorry, I jumped in too early there. All right, so yeah, so we don't lose the labels. Exactly. Right now, our idents are the cell types. So if we print them to the screen, we can see that the identities are the cell types, right? So we assign that to a metadata column. And then if we later overwrite our identities with the scRNA-Seq clusters, for example, if we look at our identities now, they're the clusters. They're not the cell types anymore. If we look at our metadata for PBMC, if we just do the $ to access the metadata columns, there isn't any place where that cell type got saved except for the place where we manually saved it. So this is why we do that, because now if I wanted to then go back and have the cell type information again, I would have lost it if I hadn't saved it in a metadata column. So exactly right, Dakota. That's exactly the reason. All right, so we can make a nice little dimension plot here, stack up our clusters side by side with our cell types. Wes says cherry keys. I'm actually not sure what that is. If you want to put that in the chat, that would actually be pretty cool, because I was actually looking at this and I was thinking there's got to be like a better way to do this, right? But yeah, okay. All right, so let's go ahead and run our find all markers command. So can anyone remind us all what this command is doing? I've learned my lesson. I'm not going to jump in too early this time. Okay, great. So Tyler says finding marker genes for all detected clusters. Yeah, exactly right. So these are the genes that are specifically, in our case, we're only looking for overexpressed. Whoops, overexpressed. So these are the genes that are specifically overexpressed in each cluster compared to every other cluster. So we can go ahead, grab them, filter them so that we only look at the significant ones, and then we can write that to a CSV file and also output it as a variable. So I'm not sure if anyone here knew that you could do that, but you can both write it to a file and return it as a variable in the tidyverse here. So we return it as a data frame, but we've also written it to a CSV. So this should have, if you're using the R projects like you should be, this should have shown up right here in the same folder that has your R projects and the rest of the code and stuff. So just to recap from last time, what I did was I wanted to say, all right, well, what's going on in cluster 0? What are the pathways that are overrepresented in cluster 0? So I just went into Excel, I filtered it, so I only included cluster 0. Did Control-Shift down to select all the 83 genes that were markers of that cluster. Went over to my browser, went to Enricher, and just pasted them. Hit submit, and now you've got your enrichment results for transcription factors, pathways, ontologies, you name it. There's 171 databases here that we can pull from. And if you recall, I took the permalink from this and I pasted it into the script. So that's actually here. So you can always come back and access this page whenever you want. It's a fantastic feature, and seriously, like, I can't give enough props to the Mayan lab. They're amazing. But let's say we want to do this in R. Well, like we showed last time, you can pick 4 databases or 5 or as many databases as you want from Enricher. You can go ahead and get your markers, pull them out as just a vector of genes, which is what we see here now, and then run the Enricher function on this. So it's basically uploaded it to Enricher, it's queried those 4 databases, and then returned the results. So we don't even have to leave R to do this. pretty nice. All right, so line 112 is using our custom plotting function that we defined earlier in the script. So hopefully you ran that. And then in line 113, we run our cow plot patchwork plot. And there was probably a smarter way to run that, because I think patchwork and cow plot can recapitulate each other's functions for the most part, but I didn't. This should have saved the result in a PNG right here. All right, so this is all recap from last time. The other thing that we could do was we could pit two clusters against each other if you wanted to really figure out what are the genes that make them different, right? So in this case, we pit 0 against 2, because if you recall, we weren't really sure why they were different from each other. They were both labeled as CD4 T cells, and it wasn't clear what made them different. So we went ahead, we got those genes that are, in this case, specific to cluster 0, because it's average log 2 fold change greater than 0. So, okay, I'll back up just to make sure that this is clear. When we pit these two clusters against each other, there's a numerator and a denominator. The numerator in this case is identity 1. That's cluster 0. Denominator is cluster 2, which means that when we look at the log fold change, anything that is greater than 0 means that it was overexpressed in cluster 0 compared to cluster 2. Anything that is less than 0 was overexpressed in cluster 2 compared to cluster 0. So just so you know how it's defined, it's easy to mess that up when you're like trying to conceptualize it, but just know it's just a fraction. All right, so we pull out these cluster 0 genes. There's not a whole lot, but at least it's enough to reasonably feed into Enricher and see if we get any results from it. We do what we just did before and save it to a plot. And that shows up right here. So be careful when you're interpreting these. You got to pay attention to the adjusted p-value. And so in this case, you can see it doesn't go above the negative log 10 p-value being 0.6, right? So it doesn't go above that. Okay, so basically a way to kind of easily figure out if this reaches significance is to actually just reverse the logarithm. So this is the negative log 10 of 0.6. So if I wanted to know what does that actually equate to in terms of a p-value, I can just do 10 to the negative, in this case, we were trying to see 0.6. And we can see that's 0.25. So not significant. Does anyone know the approximate log p-value, which is equivalent to 0.05? It's good to have, like, as a baseline, this rule of thumb. Does anybody know what it is? Okay, so I'm going to show you here, because I always use this as like a rule of thumb. So the easiest way to test is just to go log 10 of 0.05. Okay, there you go. Yeah, so Peter got it. It's negative 1.3. Yeah. So it's so close to just being 1.3 that if you're looking at a chart and you're trying to judge if something's reached significance, you can just think, did it go above the approximate, 1.3 location, I guess, either in a heat map or on an axis. Is it above 1.3? And if it is, then it's probably significant. And if the scale doesn't even include the value 1.3, let alone the value 1, probably not going to reach significance there. All right. But here we can see this pretty significant. Ribosome, very, very, very significant. So that might be the things that are distinguishing cluster 0 from cluster 2. So this is all review. Now, I asked you for an activity, but we didn't really have time to get to it, so I said I would do it now. I asked you to go and do this on the brain data set. So just to recap, this is the FACS-sorted brain non-myeloid cells from the tabula muris. Okay, so if you're using the Git repository, or at the very least you're downloading it from GitHub, this relative file path should work for you, because that should be go up a level and it's right there. So that should work for you. Okay, and then we can go ahead, run the pipeline. These are the identities we got from last time. And while this is running, I'm going to address something Wes said here. So Wes said in swear-out 3 or below, anyone copying and pasting, average log full change is average log 2 full change. So swear-out has a habit of every time they come out with a new release, changing the way that stuff is like either spelled or like the names of functions or various things like that. So if you're in swear-out 3, it should be, I guess, log full change, not log 2 full change. I'm assuming it's still a log 2 though, right? Yeah, I'm assuming it's still a log 2. Sometimes when people say log, they assume that you know that they mean log 2, which is a bit annoying. Okay. Okay, yeah, it is still log 2. All right, so we go ahead and run all that, get our identity saved. Yeah, I really don't like it when they do it either. It's kind of annoying, but oh well. All right, we get our dimension plot. So again, we did all this last time. None of this was actually part of the activity. And you can see here your cluster is now stacked up against the cell types that we thought would be in here. You know, one thing I said I was going to mention last time that I didn't is what's going on with cluster 3, which is this cluster we weren't able to tell what cell type it was. It seemed to be a mix of several different cell types that didn't really make any sense. So let's go ahead and try something. I'll put this in the chat. I'll remember to actually do it this time. Let's try something that can help us figure out whether or not this cluster is actually, I don't know the right way to put this, legitimate or not. Because sometimes when you have a hard time finding what cell type something is, it might be because there's something wrong with the cells in that cluster. So what we're going to do is we're going to actually do a feature plot, and instead of plotting like a gene or like some continuous feature that we normally deal with, we're actually going to plot this time the N feature RNA. So this will be the number of genes that had any UMI counts at all in each cell. So let's go ahead and plot that. All right, and this is actually, let's just stack this side by side with your dimension plot here, so it's more obvious what's going on. So in those cells where we said we weren't able to figure it out, these cells here, what is characteristic about them is that they really don't express a lot of genes. So there's a good chance that the reason we weren't able to classify them was because they don't really express a lot of genes, 1, and 2, because they probably aren't the highest quality. One of the things that happens when you do your clustering is that low quality cells often get put together into kind of a garbage cluster. And this seems to be one of those cases where that's probably occurring. Now, I can't say for sure. It would take more characterization and more exploratory data analysis to confirm that, but I suspect that's what's happening here, considering that there's just such a strong loss of gene expression there. We can also plot our N count RNA and see if we see the same thing, which we do to some degree. And we can also try our %MT. Oh, it's not actually included this time around because I didn't need it for anything else. All right, well, anyways, what I'm going to do here is I'm going to give you this in the chat because I know I always forget to do that. There you go. So anything that wasn't in the script, I'll try to remember to put that in the chat from now on. I'll leave it up just in case you want to see. All right. So the first goal that I asked you to do was to find the gene sets and pathways that distinguish cluster 0. So the way I started this, I just found all the clusters at first because that gives you all the data in case you want to look at something beyond cluster 0. And also it's easier to copy and paste that way. And this just takes a minute. There's more cells in this data set than there is in the PBMC one. All right. Go ahead, filter for the significant ones, save to a CSV. And you can go through the trouble of finding it in Enricher. I'm not even going to bother this time. Let's go ahead, do our Enricher in R. And we'll enrich for this here. And do our plots. And we've got here cluster 0, brain, PBMC. Pretty cool stuff. So these were supposed to be neurons, these are supposed to be oligodendrocytes, if you recall. And if you actually go on Enricher, it's very clear that you've got a strong oligodendrocyte identity in the enriched gene sets, but just none of them happen to occur in this particular grouping of 4 databases that we chose here. But what you are able to see is the other biological processes that are enriched in oligodendrocytes, such as cholesterol homeostasis, lysosome, and kind of some interesting brain regions that seem to be discovered here, and some interesting transcription factors. So if you were someone who studies neuroscience, you'd probably find this very interesting, and you'd probably understand it better. But since I don't, and I won't, I'm going to go ahead and move on now. The other thing I asked you to do was to find the genes which differentiate clusters 7 and 5, because as you recall, these were our 2 neuron clusters, and it wasn't clear why they clustered independently of each other. So I decided to check 7 against 5 here. So we go ahead and run that. Get our markers. So these are our cluster 7 specific markers because the log fold change is greater than 0, and that was the numerator. Okay. And we got 452 genes, so a pretty good number. Go ahead and upload that to Enricher. Plot and save. All right. And we see some cool stuff here. We see caudate nucleus, amygdala, some pretty significant stuff there. So perhaps that means that one of those groups of neurons was in maybe the thalamus, maybe the caudate nucleus, or maybe it was in the, the amygdala. So again, not a neuroscience person. If you were a neuroscience person, you probably understand how to interpret this. The most that I remember from I actually did my undergrad in neuroscience, so I should really know this stuff. The most I remember from my undergrad was that long-term potentiation is like how you form memories. So maybe you got something to do with that. All right, so now that we have recapped everything and I did what I promised I would do last time, which is go over the activity, I'm ready to get started with data integration here. So before I jump in, do you guys have any questions or anything that was unclear from what I just covered, which I know was super fast? So do you guys have any questions? Okay, great. All right, well, if you have any questions at any point, I'm going to keep checking the chat. Feel free to stop me anytime, and I will address any questions you have. Okay. So data integration. So this is a really fundamental topic in all single cell at this point, because you always have, in most real world studies, some need to integrate at least 2 data sets together. So what do I mean by that? Well, let's take a test case here. I've pulled a couple of data sets from a paper that I actually wrote, and these are Ewing sarcoma cell line single cell RNA-seq data sets. So this was 10x Genomics. It was a relatively recent version of the 10x chemistry, so they're pretty robust data sets. And what I want to do is compare them to each other. I'm going to say one more thing while we're running all this, but I'm going to wait for it to start running. So hopefully that loaded in pretty fast. It's very strong. It's very highly compressed so that it would fit within a smaller size for GitHub. So that might take you a minute to decompress those and actually read them in. Okay, so we're going to go ahead and run through the pipeline here. So go ahead and run this. And while we're running, I'm going to say one more thing about these cells. So Ewing sarcoma is a pediatric bone cancer, and it's actually something we study in the lab that I'm a PhD student in. So Ewing sarcoma is characterized by some really, really interesting cellular states that are in many ways reminiscent of the cell states that you find during the development of the embryo. So it's really fascinating from a developmental and a cancer point of view. Now, CHLA-9 and CHLA-10 are two Ewing sarcoma cell lines that are from the same patient. CHLA-9 was taken from a primary tumor before the patient underwent treatment. CHLA-10 was taken from a pleural effusion. So that's a metastatic site after the tumor, resisted treatment and progressed. So not to be too grim, but it's very important that we understand what these cell lines are and why we're studying them here. They're from the same patients, but as we'll see, there are some very big differences between them. Okay. So let's go ahead and plot our UMAPs here. And what we can now see is UMAPs with the cell lines from which each cell originated displayed in blue and red. Okay, so let's take a look at this and let's compare it to our clusters as well. So I'm going to also compare this to the clusters. All right, so there's some things that are interesting right off the bat. One of those things is that despite the fact that CHLA-9 and CHLA-10 are the same disease and from the same patient, and in theory, they should have at least a sizable number of cell states that are shared between them, they occupy entirely different spaces on the UMAP. The other thing to notice is that it's not just the UMAP. If you look at the clusters seem to occur entirely independently by cell line. So even the clusters, which are a more robust way of finding similarities and differences between cells, don't seem to be able to recognize these two groups as at all similar to each other. Now, as a biologist who studies this disease, I know they should be, and yet I don't see it here. So just to demonstrate that this is actually true, that they're clustering independently by cell line, I'm going to table the cell lines, which are in the original identity slot, and the cluster identifications. So tables in general, if you just run this part, tables are a way of comparing two categorical variables and then seeing the number of observations which have both, right? So the number of cells that are in both cluster 0 and belong to CHLA-9 is 350. So there's 350 cells from CHLA-9 in cluster 0, right? And so this is the distribution across all of them. And with only a few minor exceptions, they're almost entirely split by cell line. However, one of the things to keep in mind here is that the absolute number of cells isn't the best way to look at this, because CHLA-9 and CHLA-10 had a different number of total cells, which means 350 CHLA-9 cells might not mean much if it had 3 million in total, which it doesn't, but I think you get my point. So what we do is we normalize here to the total number of CHLA-9 cells. And basically, we're going to end up coming up with a percentage. What is the percentage of CHLA-9 in each of these clusters, percentage of CHLA-10 in each of these clusters? And this is kind of like a percent cluster representation. Oh, my bad here. I didn't run this yet. And so now it's the same thing, but we've got percentages instead. So we can also visualize this as a heat map if we want to. It's kind of a nice way to look at stuff like this sometimes. And you can see here, it's pretty stark. CHLA9 tends to pretty much exclusively cluster by itself, and CHLA10 exclusively clusters by itself with, again, just a couple very minor exceptions. So what's the problem here? Well, the problem is that we know there are shared cell states between CHLA9 and CHLA10, and we're not seeing that here. Why aren't we seeing that? Well, I'll make my case to you now, but basically the reason can all be explained by batch effects. The phase of the moon, the position of Saturn, what day of the week it was, who it was that ran the experiment, these are all things that somehow conspire together to make it so that if you do two different runs of single cell, they're almost undoubtedly going to look very different when you do something like a UMAP or when you do your clustering. And that's because there in single cell is a very strong batch effect, largely, I think, due to the fact that you have different dropouts occurring under different conditions. So how do we correct for that? Well, we can integrate the data. So the way that we do that is, at least this is one way, it's the way that I typically do it. The way that we do that is we split our swear-out object into a list based off of the original identity. So orig ident, and we can see here that in this case, that is CHLA9 and CHLA10. So we split it up into one cell line and the other cell line. What I do to these is I normalize them independently using scTransform. Well, normalize and scale, I guess. So this takes a minute. That took me a second. I forgot I said a planet, right? I was thinking, like, when do you use Mercury in single-cell RNA-seq prep? I don't remember that being part of the SDS. But yeah, so Wes said, don't you just hate it when you're doing library prep, but Mercury is in retrograde. Yeah, I do. All right, so we ran this. Depending on the speed at which your processor can run, it'll take anywhere from a couple seconds to a couple minutes. So if you're still running this, I'm gonna give a couple seconds to while I'm talking here for you to catch up. But if you're still running when I move on, just don't lose heart. There'll be plenty of time for you to catch up. All right, so we did the scTransform. Basically, this is a more robust transformation than the log2 and the scaling provides, right? This allows you to use the and I think I talked about this last time, but basically use the modeling method that we use for determining the mean variance trend and then regressing it away and then finding the true variable features and then applying the transformation to the data set. There's a bit more that goes into it, but that's kind of the underlying intuition behind how it's working. There's a fantastic paper they wrote all about it. And if you want to read it, I'll link it in the website for today. If you want to read it, I'm happy to facilitate that. All right, so now that we've done that, we can get our features. So these are the features upon which we will want to integrate our data sets. And the reason why we choose these features is often because when you get your standardized variants, they have the highest standardized variance. There's obviously more that goes into it, but that's, again, just the intuition. So this function is going to prep the integration. I'm not honestly sure what it specifically does. But the next step after it is to find your integration anchors. So this is basically finding places in what you could call the manifold where you should see an alignment between the different data sets that you're trying to integrate here. So we go ahead, run this. It's using something called canonical correlation analysis, or CCA, in order to do that. Again, this is pretty well described in swear-out's publication. So I would, if you want to know the math, if you want to know all the steps it's going through, I would check out those. All right, finally, we're going to take the anchors that we just discovered through that canonical correlation analysis, and we are going to merge our data sets. So it might not be obvious what we're doing here, but I think in a second, you're going to see exactly what this created. So now that we've got an integrated data set, I've called this cells.int, we can just run this right through our pipeline, and we can see what the UMAP and the clustering looks like. All right, there it goes. So the first thing I'm going to do is I'm going to do a UMAP plot for the integrated data set and the original unintegrated data set. And we're going to see what the integration actually did. So there you have it. So basically what it appears to have done is it appears to have found this donut shape in CHLA9, and then it found these other donut shapes in CHLA10, and it said, you know what, these are actually a shared cell state. And then it found the integration anchors and aligned them, leading to this now integrated UMAP, where you can see the cell states that are shared, and perhaps in the middle, some that aren't shared. All right, let's compare this to the clustering. Yeah, so now you can see how the clustering looks. So what you can notice about this is that most of the clustering is now shared between the two cell lines, except there are some clusters that appear to be quite specific to CHLA10. So let's go ahead and do our heat map again. And let's take a look at where those might be. All right, so again, you've got nice shared clusters, but you've got this one that seems to be relatively specific to CHLA9, and these two that seem to be at least somewhat specific to CHLA10. Pretty interesting. Okay. And if we wanted to do our cluster marker analysis, we could attempt to figure out what is in cluster 7. So I've analyzed these cell lines before, and actually CHLA10 has some copy number aberrations. And if you look at the dim plot, you can see basically here, rather than being just one donut, it's got some smaller donuts up here. These tend to be different clones that have different copy number variation. So I suspect that middle donut in the integrated set, where that CHLA10 specific cluster is probably one of those auxiliary donuts that's got some copy number aberration. All right, so let's go ahead and now do this. And to illustrate this, I'm going to again use a Ewing sarcoma data set, but I'm going to use one that's actually a lot more interesting than just the simple cell lines are, because this comes from a study where they used patient-derived xenografts. So let's go ahead and get the files. They should all be in your working directory right here. So these are the 5 PDXs and the count matrices for each. Go ahead, grab those, do all this nonsense, create a swear-out list. And if you recall from from module 2, when you've got a list of swear-out objects, you can merge them easily using this line here. It's a clever shortcut. I've already got these subsets pre-applied, and so we can just go ahead and run right through this. Okay, so Tyler asked, if I start out with a data file that's got cells from different patients and their IDs are stored in a ridge ident, which is actually what I'm literally doing right now, would you start out with split objects? So you wouldn't have to. Okay, yeah. So Wes says that's correct, but also when you create a merge object, you made yourself, you might want to look at the QC first. And yeah, so that's basically why I do this first, because if I had already started with it split, I wouldn't be able to, 1, visualize the batch effects as easily, or 2, have figured out what my subsets should be as easily. It's still, you can still do it, but it's probably advisable to start with everything unintegrated, do your QC, do everything else, and then determine whether or not you need to integrate things. So let's go ahead and take a look now. All right. 5 PDXs. 5 PDXs from 5 different patients in 5 different mice. But curiously, no shared cell states. Now, we know that that's not really true. We know that they have a lot in common, and they should probably be sharing quite a large number of cell states between them. So this is, again, classic batch effects, and this is what we need to integrate away. Again, no shared clusters, or at least very, very loosely shared clusters. We can show this with the heat map. So yeah, you can see it's pretty specific to the identity of each PDX. I just realized this, not everyone's a cancer researcher. If you don't know, a PDX, otherwise known as a patient-derived xenograft, is where someone takes a tumor from a patient and they implant it often under the skin in a mouse, and they let it grow there. And that's a really fantastic way for us to study it in the mouse model, where we can do things like treat with experimental drugs, which of course you can't do to humans. Okay. So this is what I want you to do. I'm going to give 10 minutes to do this, and we'll go ahead and arrive back here at 6 o'clock. Okay, and in the meantime, I'm going to look at what this conversation in the chat is saying. Okay. Okay. So, okay, yeah. All right. So, I didn't talk about reciprocal PCA. So, if you want to know about the other approaches you can use for integration, there's actually a ton of these now. scRNA, the default is CCA, but there are a lot of other approaches you can take. So many of you have probably heard of Harmony. That's one that's very popular. If you just go to the scRNA website, and we'll come over here. There's actually a large number of data integration tutorials. So there's even one on RPCA, on reciprocal PCA. But I'm really just doing the vanilla CCA integration right now. In fact, there's a lot that use neural nets, and they use autoencoders in order to integrate data. It's pretty cool. It's pretty cool stuff. So, okay. Yeah, so Wes's point is a really good one, which is if it's the same day, same sequencing run, same sample, just in different batches, all those stars have aligned, then theoretically the batch effect should be minimal. But I've personally found from experience that it can still be there. And in fact, the biggest batch effect in my experience seems to come from whose hands it was on the cells. So who was the one in the lab, and then down in the sequencing facility, who was doing the chromium capture and even just the original preparation of the tissue. So, and even sometimes it's your hands both times, and at one point you got tired. So it can be you getting tired, and then having some sort of batch effect that was created by you getting tired and maybe taking longer to do a step or something. It's part of why it's really good, if you're going to do single cell wet lab prep, to be really well rested and come in early on the day that you want to do it. I don't know. I don't do a ton of these myself. I'm mostly dry lab at this point, but if anybody who's doing wet lab prep for single cell has advice, I would be really interested to hear like what techniques you guys use. Yeah, so Wes says practice is the best for consistency. Yeah, first five were all over the place, and it takes a while to run the same cells on different days and get the same results. Yeah, it's not easy. It's not an easy protocol. There are a lot of places where stuff can go wrong. This gets particularly particularly difficult when you're talking about something like a PDX or something that's like a whole tumor or whole postmortem organs, where you have the issue of dead and dying cells to contend with. So definitely good to keep things as clean as possible at all times. So Wes mentioned that there's an algorithm that there's several algorithms for dealing with batch effects in single cell. And in fact, if you just do a Scholar search for single cell RNA, seq batch effects, there will be no end at all of articles and review papers on different tools and approaches for this. So we'll do since 2020. This is one I've heard of a lot of people using, BBKNN. And there's just really so many of them out there at this point. What's your secret, Wes? Wes has a batch screen secret. So, okay, so batch QC is your secret? Okay. I have not heard of this before. Ooh, it's in bioconductor. So this looks like a pretty nice documentation. Yeah. So let's see here. So it's kind of like an all-in-one sort of function, I guess. And it's going to do like a nice HTML report for you. That's nice. That's nice when they do that. I'll have to try this one sometime. I've heard, I think, doesn't SCVI also do batch effect correction? I think so, right? I don't remember, honestly. I've heard this one is very, very popular. But of course, I guess you do have to do this in Python. Okay, so Wes says he always runs batch QC at the beginning of a new study to figure out if you have issues in library prep, cell death, or what the problems in the wet lab are. Yeah, that's a good point. Oh, Tyler says SoupX is something that's used for looking for ambient RNA. That's actually really cool. I don't know why I just wrote that in camel case. Okay. Oh, fun. It's exclusive to oh, okay, no, they actually do have a CRAN release. That's great. Okay, that's great. Awesome. Very nice. And they got a vignette. Very nice. Okay. So this looks pretty easy to get started with. Very cool. Okay, so all right, I kind of see what they're doing here. It looks like they're finding the soup, which would be the raw gene barcode matrices where we don't yet know if they're really cells or not. Although according to Wes, we can actually figure that out on our own. We don't have to let 10x genomics tell us. It's kind of nice though that you can kind of use that as maybe a sieve of some sort. Okay, all right, so 6 o'clock. Let's go ahead and jump back in. I've been rambling enough here. All right. So let's go ahead and just go ahead and do this. All right, so the easiest way is just to copy and paste. So that's what I'm gonna do. Copy, and paste, and we'll clean this up a little bit. So get rid of that, get rid of that. This belongs here. This belongs here. And then this is basically it. Okay. Wait, what happened to 2? Okay. Well, I know where it went at least. Sorry about that. This ended up not saving me any time whatsoever. Okay. Sorry. There we go. 2. That was definitely the wording it had originally. All right, so convert cells to PDX. And I think that was it. Whoops. All right, so we've got our PDX, and we're going to split by original identity, which of course is going to give us our list containing each of the individual PDXs and the corresponding square root objects. And we're now going to run the SC transform on them. This takes just a minute. This is one of those areas where, yeah, your processor and your memory and all that is going to start to make a difference. Single cell is one of those techniques where you can do it on a laptop and you can do it on a personal computer, but there's definitely things you might want to do that start to become impossible once you get up closer to around 3,000 or 4,000 cells, or at least impossible to do in a timely manner. All right, we get our integration features, get our integration, run our CCA. So just to point something out here, when we're doing our integration, we're finding the pairwise relationship between every batch and every other batch. So the time it takes to run these steps is going to be disproportionately large compared to the number of batches you're trying to integrate. So the more batches you have, the longer this is going to take by far. I tried this once on something that had like 100 batches, and I think it took like a day and a half on a cluster. Okay, so we can integrate this now. So you can see it's trying to do everything kind of pairwise at first, and then just kind of glomming things together. All right, and now you should have your integrated PDX object. So that was task 1. Now it's done. Let's go ahead and run number 2 now, which is to just do the pipeline. Is this the right number of neighbors? Yeah, it was. Okay. Or number of dimensions, yeah. All right, done. And let's go ahead and take a look now. So we can see here our original data with all the batches. And we can see now everything has been corrected together. It's all mixed together nicely. And yet the shape, this kind of characteristic sort of teacup shape, is preserved. So you have this interesting loop here, and then you've got this larger body. And that was here, here, here. And in fact, there's one here. It's just too faint to see. I very much downsampled these data sets for the sake of time. So I know, because I've seen the full data set, that is very prominent in all of them once you have all the cells. All right, and we can look now based on clusters. We can see there's a nice mix in each cluster. Let's go ahead and confirm that with our heat map. And there you go. Nice mix in each cluster. All right. This is going into the chat now. Does anybody have any questions about that? It cut off the end of the text again. All right, I'm just gonna put that in there. Okay. All right, so if there's no questions, let's go ahead and jump into module scoring. So this is a really important component of single cell analysis, particularly for exploring your data and figuring out what's going on in the different parts of your graph and in your different clusters. So one of the things that is easiest to score in scRNA is the cell cycle. So we're going to take our integrated cell data set, so this is our CHLA9 and CHLA10 cells, and we're going to run the cell cycle scoring function on it, giving it the S features and G2M features. So the way that this is working is essentially it's taking these genes, these CC genes, updated 2019 S genes, and it's trying to score every cell in the data set to see if any of them have a particularly high score for these genes. Now, what are these genes? Well, these genes are known to be associated with the S phase of the cell cycle. So if you have a very high module score for those S phase features, you should be able to figure out if those cells are in fact in S phase, or at least predict that those cells are in S phase. We can do the same thing with G2M, and we also have G1, which is really just the lack of either of these. All right. So one thing to keep in mind is that gene symbols change over time. They change actually quite often. And so conducting this search will mean that scRNA-Seq's going to go out and it's going to attempt to find if you have like an outdated gene, or if they have an outdated gene in this list, because scRNA-Seq provides these lists. It's going to attempt to search for that and see if there's a newer version of that gene symbol that you actually have in your data set. And it's going to attempt to match these things up to the actual gene symbols you have in your data set. All right, so let's go ahead and run this. So you can see this here. It says it couldn't find any of these genes. So it's going out onto the internet and it's attempting to find the aliases for them, which are present in your data set. And even despite all that, there was some that it still wasn't able to find, which is fine. There really wasn't too many that it couldn't find. If you wanted to do this the perfect exact way, you would probably build these lists yourself instead of letting scRNA-Seq do it for you. So now we can see the cell cycle phase. Okay, so the first time I saw this, it actually kind of blew my mind. And it's the reason in some ways why not a lot of people want to do single cell on cell lines. Because I was looking at this donut for like a couple weeks. I hadn't really tried to figure out what it was yet. And it kind of all came together when I saw this. I realized the donut is the cell cycle. Because the biggest source of variance within a single cell RNA-seq data set that's based only on relatively homogeneous cell lines, the biggest source of variance is going to be what phase of the cell cycle is each cell in. So pretty cool. All right. We can also do a heat map to compare the degree to which each cell line is represented in each phase of the cell cycle. They seem to be relatively even. If we look at the actual table, we can see it's, you know, relatively even. So one of the things that comes along with doing this cell cycle scoring is that you actually do have continuous module scores for the S phase and the G2M phase. If we go to our metadata now, we can see that you have 3 new columns: S score, G2M score, and phase. Phase was what we were plotting before, where it's telling you what it thinks each phase of each, or the cell cycle phase of each cell is in. But you can also just plot the continuous values in the S score and G2M scores and see for yourself if you think it really matches up. But in our case here, I would say it seems to be pretty apparent. Okay. Okay. So, oh, there's actually a bit of a typo here. This should say hallmark G2M checkpoint. My bad there. So one of the things we can do beyond just looking for an S score or a G2M score is we can include any arbitrary collection of genes that we think are biologically important, and we can score every cell based on the degree to which they express those genes. So one of the things that we can do instead of letting scRNA-Seq do it by itself is we can actually find our own G2M gene set, and we can see how well does that stack up compared to what scRNA-Seq did for us. So one of the ways you can get gene sets is by using the msig-dbr package. It's based off of the molecular signatures database. And if you've never seen that before, if you just do a search for msig-db, first thing that comes up is this website. So the same folks who build the molecular signatures database are the same folks who build gene set enrichment analysis. They're the ones who originally published it. They're the ones who maintain it. So this is a giant collection of gene sets from all over, and there are many, many different kinds. So gene ontology is actually included in here, along with transcription factors, along with what they actually curated themselves, which is called the hallmark gene set. So those tend to be biological processes that are very well defined. It can be supported very easily by literature. So let's go ahead and grab, using msig-dbr, the hallmark gene sets. All right, so that should return a nice data frame here. And if you look through it, basically what they've shown here is gene sets and then the genes that are associated with them. And so this is the hallmark adipogenesis gene set, and these are all the genes associated with it. And then there's I think, I don't remember how many there are in the hallmark collection. I think there's maybe about 150 or so in the hallmark collection. But you can see there's lots of different kinds, and they're very well curated. If you want to learn about one of them, all you have to do is copy it, paste it into your browser. And the very first thing that'll show up is msig-db's description of what this is. So it says genes encoding proteins involved in metabolism of fatty acids, and that's hallmark fatty acid metabolism. You can learn more about it here as well. For our purposes, though, what we're going to do is we're going to take the hallmark G2M checkpoint gene set, and we're going to now, instead of doing just the bland cell cycle scoring, we're going to actually manually calculate the module score on this list of G2M genes. And we have, it looks like 200 here. So to do that, all you have to do is run add module score. So to add module score, you're going to supply a list containing your vector of genes. I don't know why they want it as a list of a vector, but they do for some reason. You can also include this search equals true. So this will do the same thing it did up here, where it'll check an online database to resolve any mismatch between the names of the genes in your gene set and the names of the genes in your data. You can also give a name to output this as in the metadata of your scRNA object. And you can also specify what assay you want to run this on. So in this case, I'm going to specify RNA, since by default, it's going to use the integrated data assay. And I don't want that because that only has 3,000 genes in it. And it also doesn't make a difference here. Okay. So you go ahead, run that. It's going to search for any symbols it thinks are missing. Doesn't take terribly long. And then now that that is done, you'll have a new metadata column. You go here, do the $, new metadata column all the way at the bottom, where it's the name that you supplied with, for some reason, the 1 after it. I don't entirely understand why they do that, but they took the name you gave it and they added a 1 after it. Go ahead, plot that. And now you can see here the G2M score as determined by your module that you manually supplied. Okay, so we can now compare this to our clusters using a violin plot. So we can see here which clusters had the highest G2M score as determined by our custom module. And then we can do a feature scatter to compare our G2M score with the G2M score determined automatically by scRNA-Seq. Let's go ahead and run that now. So pretty strong correlation here. And we can see that in both cases, the ones with the highest G2M score from our manually identified list of genes and from their automatically identified ones are both in that same G2M phase. So Ramesh asked, why don't we run it on the SCT assay? Because the SCT assay is not going to include all the genes. So when we do our transforms and when we scale our data, what we're typically doing is we are doing a scaling that is necessary for uncovering the structure in the data set. So if you're not trying to uncover data set structure, so if you're just doing something like differential gene expression and you're trying to find a module score, the log 2 of the original count is actually fine. And then you don't lose any data. I think that was right, what I said. Let me just double check that, because we could also try that the other way. It's kind of a good thing to try when you are asking a question like that. I think that you're right. I think we can also do it on the SCT and it will produce the same or a very similar result. So let's try that now. Yeah, very similar result. And it doesn't seem to have been missing any genes. So thank you for pointing that out. That was my mistake. So SCT also works. Doesn't seem to have changed a whole lot, but definitely a totally appropriate way to do this. All right. So what I want you to do now is I want you to do the same, but instead of doing this on cells and looking at cell cycle, I want you to do this with the PDXs, and I want you to do both the cell cycle scoring and also custom gene sets for hypoxia and metastasis. All right, so go ahead, complete this. I'm going to give another 10 minutes for this, and I'm going to turn off my Slack. So sorry about that. Jesus Christ. Stop. Oh my God. It's a pet peeve of mine when people don't know how to use threads in Slack. All right. So you made a good point, and this is, I guess, why I have to now hedge my statement again, which is that the actual size of the assays isn't the same. So while you guys are working on that, I'm going to have a look at this and see why I can't seem to figure out whether this is really appropriate. So let's pull out our assay object for RNA. And our assay object for SCT. And we'll pull out the data from each, because that's the column we'll be using here. All right. So this says, okay, I should have moved this over before I did that. Yeah, 33,000 by something. And then I can just do length row names SCT assay. So yeah, the SCT assay is only 18,000 something. That's a good question. So since you guys are working on this, I'm going to actually go back and show you how I would try to figure this out. Alrighty here. Where did we make this? We made this right here. Well, I'll go back to the cell one. We made this right here. So we applied our SC transform. We selected our integration features. It's not this, because then we would only have 3,000. So that's not the reason. In one of these functions, we did something that essentially caused that to happen. So I'm going to go backwards. All right. So it starts from the cell anchors here, which let's see here. So those are our 3,000 anchor features. And then we should have an object list. I feel like I'm like alternating back and forth between two buttons quickly here. Okay. Okay. All right. So at this point, by the time it gets here, it has already narrowed it down. Interesting. Very interesting. So something before this point is what does it. If this is not interesting to you, then do feel free to continue working on the actual assignment. All right, so when we did the prep SC integration, we created a cells list. And features is, again, that's our 3,000 list. Cells list, let's take a look here. CHLA9, assays, SCT, data. And again, it is a reduced set. So it's reduced by the time it gets to cells list. Cells list here. Oh no, I just realized something. I have to start back up here. All right. Whoops. Huh. So it's already there at this point. Yeah. Okay. All right. So I see what happened here. So by the time you get to here, it is already cut down to 16,000 something. So let's recap that here. So we just do our split objects. We run this. So by the time it gets to here, or sorry, that's my bad. By the time it gets to here, which I guess I probably didn't need to run again. My bad. It's already down to 16,000 or 18,000. Sorry. Come on, can do it. Okay, so by the time it gets to here, it is already at, okay, come on. There we go. Oh. Okay, so by the time it gets to here, it's already reduced. So I'm going to try something. We're going to try intersect. We're going to intersect the row names of this, because I have a suspicion as to why this is happening. Okay, I'm going to do this. No, that was right. Yeah. I don't use intersect very often, so. All right. Well, that wasn't the reason then. Maybe it was union. It was union. Nice. Ha. Does anyone know what I just did? Does anybody know how I figured it out? Anyone want to guess before I say the answer? Anyone who was following my random tapping there? Okay, if not, I'll just say, and then we can jump into the answers. Yeah, features. So features are different, except they're actually not. The same features are in both original data sets. So it's not the original data sets features that are the problem here. I know because I ran them through Cell Ranger at the exact same time. All right, so let's show this now. Okay, so there are two things that are happening here behind the scenes that you are not going to get to see. The very first thing that's happening is when you apply SC transform, all of the features that are only 0 in every case are getting lopped off. Okay, but and that's the reason that after you apply your original SC transform to cells list, you're left with in CHLA9 about 16,000 and CHLA10 about 17,000 in the SCT assay. But there's a second thing that happens when you go to integrate, which is that they now are going to take the union of the features that were in the SCT assay from both things that are getting integrated. They take the union of the two. So how did I know that? Well, I just ran this function, union. So the row names of the SCT assay data are the genes that are in that SCT assay. The row names that are in the so same thing, CHLA10 and CHLA9. I did it the same for both here. And when you take the union of them, I'm just going to show you here. Bear in mind what Ramesh originally posted. And that's the number that is in the integrated SCT assay. So that's how I know. You can also pick through their source code for an hour and probably find the same thing, but that's kind of draining. So I actually think the SCT assay might be a good one to use here, specifically because in general, you're going to have fewer genes in it that have 0 in every case. So, all right, let's go ahead and run through this. That was a good challenge question. Thank you. I don't often get stumped. That was a good one. Okay. So let's go ahead through this now. All right, so the first thing was to do the cell cycle scoring. So we can just, you know how it is. Copy and paste. Oh, I don't know why this is glitching. All right, go ahead and run that. And I'm going to put this in the chat. Okay. All right, so really beautiful here. We can see the different populations really nicely colored by cell cycle. What's really cool here is that, you know how we showed that donut in the cell line? Interestingly, you can actually see a mini donut here in the PDX. Does anyone want to hazard a guess as to why that donut is so much smaller in the PDX and why the G1 seems to be so much larger? Does anybody have a guess as to why that might be? Okay, so okay, yeah, so longer time in G1, yeah, but why? Why does a PDX have a longer time, have more cells that are in G1 than something that's, you know, in a cell line, like growing on plastic, right? Why would that be? I'm just kind of curious if anyone else like does any cancer research or anything like that. Yeah, resources, hypoxia, yeah. Just 3D environment. So imagine the 3D environment of a PDX. There's not a lot of room to physically expand, right? The center of the tumor is hypoxic and it doesn't really have a lot of room to grow. It's only the outer edges and edges that are near things like blood vessels that are able to easily continue proliferating. So there's a lot of reasons why in a real tumor in a PDX, it's actually this G1 population that might be more important. It kind of highlights one of the main limitations of of the way that we study cancer, because when you're growing cells in plastic, you treat them with a compound, you might really just be hitting the ones that are actively cycling. Most of our chemotherapeutics are hitting the ones that are actively cycling, which would be these, but you're neglecting potentially a large population of G1 cells that could come back and revive the tumor later. So, all right. So the next step, we said, okay, so we did the cluster plot as well. So we can see here how each PDX is represented relative to each phase, and it's pretty even. And we also plot the G2M and the S phase scores. Kind of nice. All right, so let's go ahead and do our cell cycle phases in a UMAP. So actually, that was what I already did here. There you go. And then it said to do the cluster heat map, which I actually already did. And that was it. So hopefully that wasn't too bad. The next part was the module scoring and comparison. Now, this part I expected to be pretty difficult. So the first thing was to get the hallmark hypoxia gene set, which I actually gave you the code for that already. Now I wanted you to calculate the hypoxia module score. So just like up here, we're going to take our add module score command, and we're going to go ahead and do that here. So I just called this hypoxia score, and we'll do the SCT assay, and we'll give it the hypoxia genes to work with. And then, of course, we're dealing with the PDX here. So copy or replace that. And then just the final touch is there has to be a 1 at the end. Again, I don't know why. All right, go ahead and run that. This will take a second because it has to find quite a few genes that are missing. One of the disadvantages of using SCT is there's actually more genes missing in this than when I ran it earlier using just RNA. But that's okay, because they're genes we didn't really care too much about anyways, I'm guessing. To speed this up, you can turn search to off. So you can put false there. All right, let's go ahead and plot that. All right, so nothing super stellar, I guess. But yeah, actually, this looks a lot worse than it looked when I was doing it before with the RNA assay. But anyways, so yeah. So you can see here where the strongest signal is probably down here. And I asked you to then compare two clusters for hypoxia score using the violin plot. And so we can do that here with a VLN. Hopefully you remember how to do that. Starting to give you stuff that I'm not immediately letting you copy and paste easily, because this was actually something we did in module 2 and 3. All right, so we'll do hypoxia score here. And then we will do group by square up clusters. So interestingly, it looks like cluster 5 of all of them seems to be the most hypoxic. If I was going to dig deeper into this, I would probably try to figure out what else is going on in cluster 5. All right, so the next one was to do the same, but with the metastasis gene set. So this is pretty easy. We're just going to replace hypoxia with metastasis. Oops, that's not what I meant to do. All right. Oh, I think I called them EMT genes. Probably should have called them MET genes or something that would have made more sense. All right. Find. Yeah. Okay. I gotta run this still. Gotta run both of these still. Yeah. All right. Sake of time, I'm going to skip that. And I'm just going to skip the search. Because I know that the vast majority of these are, it's searching for them because they're it's not that they're not in the data, they're just not in the SCT assay. Because I've run this before. All right. And it looks like there's kind of some interesting pattern here. I'm not sure why. This doesn't look quite right compared to what it looked like before. Maybe there's some reason to use the RNA instead. Well, I'm going to go back and I'm going to do it the way I did it originally. So at the very least, you can see what I was expecting to see and what you will probably expect to see here. And at some point later, I'll try to figure out the reason to use one or the other. And then I will let you know what that is. All right. So yeah, this one's a bit clearer now. I know it doesn't look too different, but it is different now. Okay, so let's go ahead and do our violin plot now. So nothing super clear here. Pretty interesting, though. There's a few that seem to have higher median levels. We'd have to do more analysis to really see what's going on. Finally, I said as a bonus to do a feature scatter to compare hypoxia metastasis scores in clusters 2 and 5. That was a big ask. Does anybody have the command that it would take to do that? Does anyone want to put that in the chat? Does anyone get this part? You can do it all in one line, actually. Yeah, I know this was a pretty tough one. Okay, so here's how you do that. I'm actually going to go back and put this back into RNA here, just so it's consistent. All right. Okay. So the way that you would do that is with a feature scatter. Okay, so that's the first part. It's going to be a feature scatter, and then you supply pdx.int. Feature 1 will be our hypoxia score 1. Feature 2 will be our metastasis score. Now, how do you get these cells that are only in clusters 2 and 5? Interesting. So those of you who might have seen, there's an argument here for cells. So I can tell it which cells I want to include. But how do I know which cells are in those 2 clusters? So you might remember this from a couple weeks ago when I showed you how to do this. But the way you can do it is with cells by identities. And so what that's going to do is it's going to grab the cells that belong to some particular identity and return them as a list, which you can then unlist. All right. So object is pdx int. Idents will give as 2 and 5. So if we just run this part by itself, we'll just run this cells by idents inner paren. We can see here it's found for cluster 5, all the cells, and for cluster 2, all the cells. And then when we unlist this, it'll return it as a vector. And then we can supply that to feature scatter. All righty. And there you have it. So we've got our clusters 2 and cluster 5, and we've got our scatter plot showing the relationship between hypoxia and metastasis. This is something that has been talked about a lot in the cancer field, whether there is a relationship between the hypoxic component of a tumor, the inner hypoxic component, and the ability to start metastasizing, because we know hypoxia might lead you to want to get more oxygen and therefore bring in blood vessels, and therefore you now have the avenue to escape the tumor and go somewhere else, right? So there might be a relationship between these. It might be weak, it might be something, but we can see here there is a potential correlation here between these. Okay. And that obviously, if you were going to make a statement like that in a paper, you would want to really, really back it up, which I'm not doing here for the sake of time and also for the sake of the fact that that's not what I'm studying right now. Okay. So before we jump into our last topic, which is pseudotime, I wanted to ask really quickly, are there any other questions? Anything that I went over that seems unclear here? Any questions about why we would do this, or what the benefit of doing something like this provides? I'm going to make one note that I forgot to make earlier. Oh, and I'm going to put this in the chat. Let's go ahead and do that. Okay, well, it got down to the add module score part, which is nice. So I'm going to start from here. There we go. So that should be all of it now. The other thing I didn't talk about is integration, right? So cell type identification using integration to a reference. Okay, so just like we integrated our different batches from our PDX, just like we integrated our different cell lines from our Ewing sarcoma cells, you're able to also integrate a query to a reference, right? So you can take, let's say that you wanted to take your PBMCs. If you already had a pre-labeled PBMC data set where you already knew what all the labels were, you could integrate them together, the unlabeled and the labeled, and then transfer the labels from the labeled data set to the unlabeled data set. And that is, in fact, what they will show you how to do using a new SwearOut4 feature, which is mapping and annotating query data sets. And there's an entire tutorial on how to do it here. I didn't go over it because it's just not something that I think most people are trying to do yet, but I think it's a great way. And it's actually something I'm using right now. And if you want to see how to do it, you can go here. I'm also happy if someone wants to see how to do this to show this in the next week's, or I should say in the last week's presentation. Ramesh asked, is there a way to add phase assignment onto a feature scatter plot using two different shapes? Shapes? What do you mean by shapes? Like instead of these being points, they would be like triangles and squares and stuff? Yeah, I mean, you can do okay, so let me just show this here. So you can do here group by phase, and now it's colored by G1, G2M, and S phase. Is that what you were referring to? Yeah, then you lose cluster identity. Yeah, that's definitely for sure. Okay, so I know what you're asking because I tried to do it earlier today and it just did not go very well. So what you can do, there is an option to do a shape by. Wait, I don't think that was the one I was looking for actually. Well, okay, it says it's ignored, but I know it does something. Yeah, Wes's answer is probably the best one. So yeah, you can see here it's now trying to like make them different shapes based on the phase of the cell cycle, but it looks terrible and you can't really see the difference. Yeah. Ultimately, I don't see. I know, Wes, you said you like swear-out for its plotting, but I actually don't care at all for swear-out's plotting because that you lose all control really over the plotting side of things. Now, one thing you can do, and I think you were alluding to this, is you can say my plot. You can export this as a ggplot because my plot is a ggplot object. So if you wanted to, and I'm not even sure how to do this to like a plot that's already got stuff on it. Does anyone know how to do that? I don't know how. I don't know how to go back and retroactively change the aesthetic mapping because the aesthetic mapping was already set by the feature scatter command. But anyways, I guess you could, in theory, go and do mapping equals AES shape is, I don't know, phase. Yeah, I didn't think that was going to work anyways. Yeah, all right. I'll try one more thing, and then this will have to be something that goes like in the thing later. Yeah, that is not working well. Okay, never mind. All right, so Wes says he'll send it later. Okay, that would be great. That would be great. Yeah, because my instinct is frankly just to take the metadata from this and do all my plotting completely custom in ggplot instead of trying to wrangle something out of their swear-out's pre-made plots. All right, so final part, which is actually going to go relatively fast, is pseudotime analysis. All right, so pseudotime is a concept that is basically the idea that your cells are undergoing some sort of continuous transformation, right, or continuous change. So let's go ahead and look at that in the context of our cell line, because that's a great test case for a change that's occurring based on something continuous, which is, of course, in this case, cell cycle. So, okay, so I guess I forgot to include this, but you need the library swear-out wrappers. So that was one of the things I had you install earlier if you installed those two libraries, but I didn't include the load function. I assume most people weren't going to really want to try to do this because it was really hard to install. Okay, so go ahead and convert this. Now it's going to throw a warning message because basically you've converted to the monocle 3 data type. So you're converting out of swear-out and into monocle 3. So for those of you who don't know, monocle 3 is just it's another pipeline that you can use for processing single cell data, but it includes trajectory analysis, which swear-out does not include. All right, so one of the things you have to do is you have to recluster your cells using monocle because they're actually going to save some information you need for your trajectory. So we go ahead and run line 454 here to recluster our cells, and it's done. Great. Now we're going to learn the graph. This is where they're going to learn the trajectory based on the graph. It always tells me this. I really don't think it means anything, really. I've never found it to be an issue. All right, and now finally, we get to see our cells in UMAP with our trajectory overlaid on top. So we go ahead and run this line 460, and we'll see. And there you have it. That's a trajectory. So it might not be immediately apparent what's going on here, but I'm going to show you how we can actually see this more clearly using our pseudotime ordering. Okay, so basically, this is the trajectory that we found, and then pseudotime is where we apply a value or where we find a value for every cell that represents its position along this trajectory. So if you imagine this like a garden path, you start here at time 0, and then you walk all the way to the other side. Well, the time that you take to walk to each individual point along that path gets recorded. The final time will be the entire time it took you to walk the whole length. And then the starting time will be 0. Okay. So what do we think this is representing? Well, we can take a look at our phase, which again, we know is the biggest source of variance. And we can see here, this looks suspiciously similar to where we saw our trajectory. So let's go ahead and order this by pseudotime. So we're going to get that pseudotime value for every cell. And to do that, we can actually use a cool trick here using the order cells function. And the way this is going to work is you're going to run this, and it's going to pull up a Shiny app in RStudio. Really cool. And it's going to ask you to pick the start nodes. Okay. So let's imagine you had a much more complex, right? A more complex trajectory structure. You'd probably have a couple places that you would consider to be start nodes or like basal states. But in our case, we can do this pretty easily by just selecting this one right here. And I think you'll see in a second kind of what this is doing. So we choose this, it highlights it red, and we hit done. And now it has finished ordering all the cells based off of where we said the start point is. All right. Now let's go ahead and plot the pseudotime with the trajectory on top. Okay, and there you go. So pseudotime, the place that we started is going to be 0, and the place that we end is going to be the final value, which in this case was about 15. And so you can see here, the pseudotime for every cell is displayed. Now, all we really wanted from this analysis was to get our pseudotime scores back. So let's go ahead, take that pseudotime, add it back into our original swear-out data set using this line here. And one more thing that we are probably going to want to do is take all of the infinite pseudotimes and set them to NA. Can anyone tell me where in this graph I might have infinite pseudotimes? Does anybody want to unmute or try saying in the chat where you might get an infinite pseudotime? So, no, not the center, not the center. So recall that the pseudotime is the time it would take to walk from the starting point to any other place on the graph. And at some point, you can walk to everything on the graph because everything is connected to each other by the same trajectory. But there's a few cells that are not part of this graph. Yeah. Okay, so Tyler got it. Yeah. These cells over here are not part of our graph network, right? So recall that like sometimes you can have disconnected components of the graph. No matter how long we walk from this point, there is no way we can get here. There is no path that leads us here, which means that the pseudotime, the time it would take to walk here, is literally infinite because it's impossible to get here. So pretty cool. But if you have infinite values in R, that can really mess with some of your other functions. So I set these to NA. All right, so let's go ahead and do our feature plot now and show our pseudotime score. Oh yeah, I forgot to run this line. My bad. There we go. So pseudotime score like this. And then yes, these were the ones that are NA, which is why they're gray. So we already kind of alluded to this, but this lines up with what? Well, it lines up with the cell cycle. Oh, shoot. My bad. This should have been dim plot. Kind of annoying that you have to like specify dim plot versus feature plot, but it's fine. Group by phase. Well, that was not what I wanted. Doesn't want to put them side by side for me, I guess. Sorry about that. Come on. There you go. So what's really cool here is that this pseudotime boundary is right at the boundary of the G2M to G1 mix here. So what this implies is, in fact, this is not really a donut. I mean, it represents it as a donut, but there is no clear connection between G2M and G1 here. There is G1 to S to G2M, but there seems to be some sort of barrier here between these. Now, in our way of seeing the world, obviously you can get back here because we know how biology works. But in the world of this graph network, there wasn't a clear connection to get back here, because even though we can learn nonlinear relationships, UMAP is not very good at learning circle relationships. So it's not really good at learning that this is actually a connected circle with a cycle in it. And the pseudotime is definitely not good at that. Trajectory analysis is definitely not good at that. So when we do this graph construction here, even though UMAP does embed this as a circle, and even though the graph does contain a circle within it, the trajectory analysis does not have the ability to recognize that you're actually going back to where you started once you get here. So kind of interesting. I don't know. Kind of highlights how each different approach makes different assumptions. All right, so now I want to do a pseudotime analysis on the PDX. And this is going to be the last part where really I'm just showcasing this. I'm not going to ask you to do this as an activity because I didn't really want to make you install all of this stuff because that was a huge pain. I'm going to put this in the chat. Okay, so let's go ahead and do this with a PDX now. So we do our PDX, we get our clusters, learn our graph. I'm gonna, just for my purposes, I'm going to put the phase here again. There we go. So we can see where the phases were. And now let's select some start points. Okay. So we can see here that in our representation of the data in UMAP, they found a few different nodes, places where there could be trajectories starting or ending. And there's actually some really interesting patterns here. So let's go ahead and, oh, I guess I already did have this. There we go. Let's go ahead and select some start points. Oh, when I did a find and replace, I messed that up. All right. So it's now asking me to find my root nodes. Okay, so my root node, I believe that my roots are going to be here because this is where we have cycling cells. And I think that they undergo a transformation once they get to the center of the PDX. And maybe they go off on these two different branches. Maybe these are like leading edge. Maybe they're still hypoxic, but maybe they're starting to become invasive. And maybe these are true hypoxic and they're just totally stressed. It's honestly hard to say. This is one of the reasons that not everyone does pseudotime, because it's actually pretty hard to interpret this. Made the same mistake here. All right, so let's go ahead and color this by pseudotime now. I'll correct this for the final. All right, so see here, pseudotime 0 down here, and then we're going in, we're finding these interesting pockets down here. You could have also selected a couple of nodes, and that way you could have had a more interesting pseudotime analysis. So if you wanted to see how are these different from each other, you could have selected this as the root, and then you could have figured out how these are different to some degree. All right, let's go ahead and add this back in. And now we can see our pseudotime scores here. Okay, so I don't typically use pseudotime analysis. Not a lot of people I know do, but in case you wanted to do it, because I know it is a popular technique in the literature, I wanted to show you how you can do that with staying in the swear-out environment and just kind of doing things in Monocle on the side using the swear-out wrappers package. And that's it. So the next time I'm going to talk about the projects that I promised that we would do. And this is optional. It's if you want to do a project and have someone give you critical real world feedback, you'll get to do this and I'll do that for you. And I'll also talk about the discussion of missed topics. So the project you can take as long as you want to do. As long as you were in the workshop, I'm happy for you to be doing these projects, and then we can meet up. I've got my office hours posted. Those don't change even after the workshop ends, and we can go over it together. And then we're also going to do the final discussion of any topics that we missed that you wanted to go over. So send me your requests for topics if we didn't cover some that you wanted. And I'm going to see if I can convince Wes also to give a presentation for next time. So that's gonna be it for now. So do you guys have any questions? Anything else that you wanted to ask that I didn't get to? We're ending early. I feel bad. Well, not really early. Earlier. Okay. Well, I'll stick around for a couple more minutes, but if you don't have any questions, feel free to head out and I will see you next time. But I'm going to stick around for a couple minutes just in case you have anything that you want to ask. Okay. Yeah. So yeah, do feel free to head out if you don't have a question. But yeah, I'll stick around for another minute. I'll stick around for another minute if you've got any questions you want to ask. Okay, so where did the PDX data come from? So you could only find the geo-accession numbers. So I had the data preloaded. You shouldn't have needed to go out and grab it from anywhere because I actually made a downsampled version of those data sets. Or are you asking just like if you wanted to find those, how could you find them? Okay, so let's see here. So I grabbed this from this paper. So anytime you're reading a paper and you want to reanalyze their data, one of the things you can do is you can, I'm going to show you legitimately, if I was in this workshop right now, how I would have figured this out. Okay, so what you can do is you can come over here, go to that paper, and if it is in a reputable journal, it is going to have a geo-accession or an SRA accession or something. If you just, anywhere here, if you're looking at the full thing and you just do a find for GSE, GSE, actually we'll do for GO, okay, this is, I guess I used the word GSE too many times. Sometimes you'll pull up GSEA when you do this, which is kind of annoying. I wrote this paper too, I should have known better. All right. Wait, where are my data sets? I included that section. Oh no, that's not good. Ooh, I'm gonna get in trouble for that. Oh no, there it is.
No, it's there. It's there. These are fine. I got those. I was just mixing it up. I thought that I was supposed to download also this my matrix CSV, but that's the downsampled one. I got it now. I was just mixed up.
Okay, gotcha.
Sorry.
All right.
Yeah.
But yeah, but if you do want to find from a paper, if you do want to find the geo-accession, you can just search GSE until it pulls up the data and software availability section.
Yes, sure.
Okay.
Thank you.
All right. Well, thank you all for hanging out. And yeah, send me your questions if you want me to go over anything for next time. And then otherwise, I'll see you all in a week. All right. You all have a good time. Okay.